My topic is entitled as Natural Convection of Rigid-Free Nanofluid Layer in High Porosity, Jeffrey model. This is the abstract part. This study is based on the two major factors, Brownian motion, random movement of microscopic particles suspended in a liquid or gas, such as the continuing erratic behavior of dust particles in still water. The movement is produced by the natural kinematic activity of the molecules of the fluid that strike the foreign particles, also called Brownian movement. And thermophoresis. Thermophoresis is a phenomenon observed in mixtures of mobile particles where the different particle types exhibit different responses to the force of the temperature gradient. A Doppler effect is the heavy flux due to mass concentration gradient occurring as the coupled effect of irreversible processes. It is the reciprocal Surender effect. Introduction, non-Newtonian fluids are used in different industries and find important applications in various fields of science and technology, such as manufacturing of plastic, polymer industry, dyeing of papers and textiles, food processing, geophysics, and biological sciences, with the relevance of non-Newtonian nanofluids in modern science and technology. Various constitutive relations have been derived in the modeling of non-Newtonian fluids. One such type of constitutive relation is the Jeffrey non-Newtonian nanofluid. Jeffrey model is linear model that uses time derivative instead of convective derivative. Keeping in view the various applications of viscoelastic nanofluids specified above, the main aim of this paper is to study the effect of the Jeffrey parameter. These are the applications where it is used: food processing, filtration, oceanography, limnology, geophysics, engineering, and soil sciences, etc. This is the mathematical problem where Jeffrey model is used. Heated from below, the problem is taken. These are the governing equations which are expressed in 1, 2, 3, and 4. Equation of continuity, equation of momentum, equation of motion. And these are the known additional parameters: thermal diffusivity and thermal capacity ratio, Prandtl number, Darcy number, Wadhams number, nanoparticles number, and modified diffusivity ratio. And these are the perturbations which are used here. And the analysis, normal mode analysis, and Clarkin weighted residue method is used here. After using these methods, we got the Rayleigh number, which is expressed in equation 24. And then we put omega is equal to 0 in the Rayleigh number. Then we find the Rayleigh number for non-oscillatory convection, that is natural convection. Numerical results and discussions. So here we find the effect of lambda, that is the Jeffrey parameter. When we give the different values for the Jeffrey parameter, we find the Rayleigh number increases and the system is stabilizing. Similarly, we have found the impact of lambda, that is Jeffrey parameter, on top heavy configuration. Here you also we get the different values for the parameter. Again, the system is stabilizing for the top heavy configuration. Similarly, for the Darcy-Brinkman model, I have found the impact of this number. It is stationary. There is no stabilization and destabilization. And the conclusions: Jeffrey parameter, noise number, and modified diffusivity ratio stabilizes the physical system. Medium porosity stabilizes the onset of stationary convection. Jeffrey parameter advances the onset of stationary convection. Stationary convection is independent of both Prandtl number and particle density Rayleigh number. And Limitations: the current exploration of the impact of rotation on thermal instability in a dusty Brinkman porous sphere filled with a Jackson nanofluid layer has several limitations. It often relies on simplifications and assumptions that may not fully capture real-world complexities. There is often a shortage of comprehensive experimental data to validate theoretical models. Future scope: in this study, we investigated the influence of rotation on the thermal instability of a Darcy-Brinkman porous layer filled with a Jeffrey nanofluid. Our research serves as a foundation exploration that can be extended to examine the effect of magnetic and electric fields with potential applications across various engineering industries. References: these are some references which are used here. Thank you.
Yes. Now you can put on slide mode. Yes. Perfect. So you can start.
Good afternoon to all the participants of this conference. I am grateful to the organizers for giving me an opportunity to present my work. Today, I'm going to talk about portable and affordable paper-based analytical device for on-site detection of perchlorate in contaminated water. Just to introduce myself, I am Dr. Ashish Kapoor. I'm a professor in Department of Chemical Engineering at SBTU Kanpur. My research area involves development of low-cost microfluidic devices for which have applications in environment, food testing, as well as in biomedical diagnostics. So today I'm going to present the work which we have done with respect to the detection of perchlorate in water contamination. So the outline of the presentation is shown here. First, we'll just briefly discuss about the importance of the detection of perchlorate, how it is contaminated, why it is important to detect it, then what is the role of microfluidic sensors in detecting this contamination,
With
and
presentation, Tina.
how we
Oh,
fabricate these paper-based devices for detection of perchlorate, and then the studies which we have done, including the real sample analysis, a brief discussion on that, and then analytical performance evaluation of these devices, and then the conclusion. So perchlorate, it is a chemically hazardous compound which is composed of chlorine and oxygen atoms. Now it is comes into the environment in the different ways. One of them is it occurs as a natural impurity in the salts which are used in producing the fertilizers and similar products. But the other major component which comes is from the industries which are involved in the manufacturing of the explosive, firecrackers, and such systems, because it is used as an oxidizer in these kind of systems, and it finds its pathways from there to come into the environment in different matrices. So the main exposure for humans to perchlorate is in form of the ingestion of the contaminated food and drinking water, and it affects the health in different aspects. Some of the major aspects are shown here. For example, the thyroid gland in the humans and the other animals is the primary target of this perchlorate toxicity, because it can interfere with the iodine uptake, which with the into the thyroid gland when the concentrations are beyond the acceptable limits, and it disrupts the functions of thyroid and lead to the reduction in the production of the thyroid hormones. And it also affects the hormones which are important for the growth of the central nervous system, especially in the newborn babies and the pregnant women and so on. So it is important to come up with the techniques by which we can effectively detect it, and so that the appropriate measures can be taken to curb it. Now, conventionally, there are many techniques which are used for detection of perchlorate, but all of them have some kind of limitations, because of which there is a need to go for the alternative sensors. Of them, I'm going to talk about the microfluidic sensors. So just to give an introduction about the microfluidics, what it is about. It is the science and technology of the systems that process or manipulate the small amounts of fluids using miniaturized channels. So when we are talking about the microfluidic devices, we refer to small scale devices, something which you can hold in a hand, and it needs very small amount of volumes. That means to the order of nanoliter, picoliters or below. The dimensions in these devices, the channels which they consist of, are typically of the orders of nanometers, microns, and so on. So these require very less amount of the energy to transport these fluids. And sometimes we make use of the passive flows also that will differ. And not only are these miniaturized devices, but also at these scales, we have importance of some phenomena like laminar flows, faster kinetics, faster heat transfer and mass transfer. which plays to our advantage in developing these devices. So for all these advantages, they have gained a lot of applications in the analytical domain because we need very small amount of samples compared to the conventional devices. And so if you're using some reagent which are costly, you can save on the cost of them. And we get much faster response. And these are also low cost devices. And now among these microfluidic devices, a particular subcategory which I'm going to focus on is the paper-based devices. One of the main advantage of this paper-based device is that these are made up of the intrinsic capillary kind of a substrate, capillary induced substrate. So whatever flows occur, they will not require any amount of external amount of pumping. So we can avoid the pumping cost as well. So, and also since they are made up of the cheap substrates, this brings them the cost and also gives them flexibility to the device itself. So nowadays, a lot of talk is going about the development of the wearable and the flexible devices for which these can play a very important role. And also because of the nature of these substrates, they are also biodegradable. So it is environmental friendly. So considering these aspects, many 2D and 3D fabrication techniques have been developed to develop these devices, and many flow control strategies are developed so that we can manipulate the amount of the fluid, how they are flowing in it, and we can get the desired outputs. So with this, lot of developments are taking place in the area of the health diagnostics, environmental monitoring, food quality control, drug analysis, and cell assays. Now, these rely on various types of fabrication methods. Some of them are derived from the conventional electronics industry when they use the lithography and so on. But more recently, the alternative and cheaper methods are coming into picture. For example, screen printing, laser printing, wax printing, and so on, by which we can fabricate these devices in a very cheap and easy manner. And they have found applications in the many areas of the public and societal relevance. So I'm just going to tell about one of the techniques which we have adopted and in our studies, that is a wax printing. Now, it is a very simple method. The way it works is, suppose you take a piece of paper, like a filter paper, which we use it typically in our labs. And as it is shown in the figure, if we just pattern it with the wax, for example, which is shown in the black kind of arrows here. Now, when we heat this paper, what happens to the wax is it will melt and it will permeate through the porous matrix of this paper substrate, as it is shown in the third one here, that the wax melts and spreads throughout. So whatever is left in the between these patterns, that is hydrophilic. And now once we remove this pattern paper from the heated substrate, like a hot plate, then wax will again become converted into the solid. So what ultimately we get is, we get the hydrophilic regions which are surrounded by hydrophobic boundaries. So this serves as our test zone. So if we put any amount of liquid, it will remain confined within these hydrophilic regions. These are shown as a test zones here. So this can be done in any lab without any need of any sophisticated facilities. All we need to make is the pattern design, which can be made using simple softwares like PowerPoint itself. So we use a slightly modified version of this so that we don't even reuse the drawing. We use the stamp mediated wax printing, in which we made a customized stamp, which we use in our daily offices. We gave it the design of the stamp such that we can get the required design of the test pattern on our final substrate. So we took the molten wax and we just put our stamp in the molten wax, and then we stamped it on the filter paper, and then we allowed it to cool. We heated it so that it went into the inside the paper substrate, and then we allowed it to cool, so it formed these boundaries. So we got a well-defined test patterns, test zones, which became our test regions. So using this technique, we fabricated this spot test type of devices. They can also be made into the flow type of devices. And then we use the reagent methylene blue, which reacts with this perchlorate and forms the purple colored complex, which indicates the presence of the perchlorate. And we did this for different concentration ranges. And we use a smartphone to quantify how much is the amount of this purple color which is obtained in each of the test zone. And then we converted that color into the color intensity, which can be just by converting each of the color. As we know, it is composed of RG and B, and that can be converted into a holistic grayscale intensity. With the help of that, we get a calibration plot for the grayscale intensity versus the concentration, which can be used for quantifying the amount of the perchlorate which is present in the contaminant. Now, this device was used for testing the real samples which were collected from various districts in Tamil Nadu. Uh, so some of these areas were chosen because some of them are house of some of the firework industries, and it has been known in the literature that perchlorate contamination is present in the groundwater bodies there. So using our device, we tested this contamination, or so we tested about 30 samples on about 12 of them gave positive response in terms of perchlorate. And not only we got the qualitative output, but also we got the quantitative output. How much is the perchlorate present? Now, to quantify the such sensor, it is also required to perform the various other tests, including the interference test. So we tested several other ions which can be present along in the water, whether they will interfere with the testing of the perchlorate or not. So all these individual ions were tested, and then they were also mixed with the perchlorate solution and tested of the known concentration. And the results were quite promising that the device was not susceptible to the interference from the other parameters. Further, we also tested the stability of the device. For example, suppose we take the measurement and we take the picture of the my perchlorate presence. Then what happens if I leave this device for a certain amount of time? Will the signal be same or it will deteriorate further? We tested it for 24 hours, and the deviations were within the 10% within 6 hours, and after 6 hours, then the signal started to deteriorate. So usually 6 hours is more than enough time to get the sample images. So this tells that this device is pretty stable. And also we investigated whether this particular reaction that we have done, it is specific to the perchlorate with the methylene blue or not. So the signal which was obtained with respect to methylene blue was much higher compared to the other device that we used. So it was very specific, and we persisted in continuing with the methylene blue based devices. So overall, we have demonstrated a simple device, which is a cost-effective paper-based device for the rapid and precise detection of perchlorate in water samples. So the device was designed and fabricated in this work as a rapid screening tool for detecting the perchlorate in the water samples. This was also investigated for examining the stability and specificity of the assay. And we also combined the smartphone-based techniques for colorimetry detection so that it can be used for on-site detection of the contaminants. So overall, this study contributes to the development of an innovative and sustainable method for quantitative detection of perchlorate. And this can be extended for many such contaminants and for the water quality monitoring applications. So thank you all for your attention. I'll be happy to take any questions on this.
Good afternoon, one and all. So I'm Dr. Nagendra Prasad, Assistant Professor, Department of Chemistry, SJC, JSSSU, Mysore. Today I'm going to talk on Citral-Tryptamine conjugated silver nanoparticle and their application towards antibacterial, anti-cancer, and sensor. So in this present study, we synthesized a citral-based imine molecule and undergo some spectral studies like NMR, FTR, LCMS, and we got a good result and good agreement with the molecular formula also. And we investigated some biological properties by in silico DNA studies via Q and docking analysis. And we can find out docking analysis, interactions and binding affinities using different protein IDs. Again, to enhance the antibacterial and anti-cancer activities of particular compound, we are going to synthesize silver nanoparticle using the citral-tryptamine based compounds. Again, that silver nanoparticle get analyzed by XRD, means some analysis. We confirm that nanoparticles. Again, that nanoparticles will subjected to antibacterial and as well as an anti-cancer activity. We got a good result. And finally, we are going to test it for sodium nitrate sensing. Also, we got a good result in case of sensor also. So look at the scope of the present work that is synthesized some organic compound like containing citral and tryptamine. It shows very good bacterial activity and as well as an anti-cancer activity. So we did DFT studies and also we did docking analysis for the particular compound. And we using that compound, we are going to synthesize that silver nanoparticles. And that silver nanoparticle also enhance their activity that will prove. This is the scope of the present work. And the right side is a synthesis of citral-tryptamine based silver nanoparticle. Look at the schematic representation of that synthesis. First, you synthesize the imine-based compound, then imine-based compounds again treated with an silver nitrate, so in the presence of L-ascorbic acid. So you got in silver nanoparticles. That's it. Before going to synthesize that silver nanoparticle, so we did some in silico studies against citral-tryptamine based compounds. So you got good result, good interactions of that compound against a particular protein ID, like 6S ZA and 3M2W and 3DAQ and 680M. It's with respect to what antibacterial and anticancer studies. And then I compared to the standard ligand also, you got good result in the binding score. You got minus 6.62 in case of protein ID and minus 8.11 for the standard ligand. Compared to that standard ligand, you got minus 6.55. Like that, you did DFT analysis also. You can find out band gap, electron band gap using that DFT for the CT compound. Then we analyze that ADM analysis and the bioactive score using in silico studies. So you got good result for 99.4% observation ABS in case of ADM studies. And in case of fast analysis, also you got the compound as a moderately active using that probably active parameters. And boiled again GI representations, the point indicates that point. So in between yellow region, that indicates what it's very good activity towards the antibacterial and antibacterial activities. And then look at this bioactive score. It is an nice kinase inhibitors and nuclear receptor ligands. You got moderately active. From these studies, we conclude that our compound, CT compound, has moderately, and so one ADM studies shows that good activity towards what antibacterial and anticancer studies. So characterization of CT at ADM after synthesization of a silver nanoparticle, we did characterizations of silver nanoparticle like UV spectra and XRD and SEM analysis. UV spectra analysis, according to the literature, UV spectra of silver nanoparticle comes under 450 nanometers. We got at 450 broad peak 450 nanometer. That will confirms the synthesized particle is silver nanoparticle. Then XRD analysis also, according to JCPDS value, our synthesizer compound shows a very good agreement with JCPDS value. And SEM analysis also, we got plate-like structure in case of SEM analysis. And finally, we did for EDAX analysis, you got a silver, carbon, and nitrogen in case of EDX analysis. And then we can confirm that our compound containing silver nanoparticle along with the CT compounds. Then subjected to biological activities, you got bacteria like Staph aureus and Bacillus cereus, but it is in comparatively that gram-negative, gram-positive bacteria against gram-positive bacteria, our compounds have very good activities. These are all the picture representations of the activity towards anti-MRSA. So then membrane damage assay for particular compound for AG at CTNP. So SEM analysis shows that membrane damage. Look at this figure 8 and A and B. So this one will indicate some rupture in case of a membrane. That indicates in case of SEM analysis, we can confirm that. So our component will act as an antibacterial agent. Then in another activity that will provide an evidence for membrane damage assay, that will is in cellular content leakage and measurement of potassium content. They look at these two graphs that indicates the increasing in the absorbance and increasing the concentration of potassium in the media. So while treating that CT at AG and P against a particular bacteria, that indicates membrane get damaged. This is also one of the assay to validate the membrane damage assay, that is what using what cyclic voltagram. The release of an electron towards that indicates the peak indicates that release of an electron while treating that CTAG and P against an MRSA bacteria. The graph here shows that there is no peak of the glucose and pure culture. There is no rupture in the membrane. So another assay I can conclude that anti-cancer activity of CT8 AGNPs, that AC50 value got 100 microgram per ml. So that figure 12 indicates that A is an SICH cell line control, and B it is an after the treatment of CT8 AGNP. It is in, the look at the A and B comparison, we can conclude that our compound will act as a good agent against SIH cell line. So another application towards the sensor applications of sodium nitrate, against sodium nitrate, our compound, that the look at the graph, figure 13a indicates that our CT8 AGNP shows the oxidation and reduction peak. It is a very good, it will act as a very good electrochemical sensor. So, and cyclic voltogram with a different scan rate, we can validate that our compound will sensing that sodium nitrate compound. This is another method, LSP method. We can find out the CT8 AGNP at GC, the presence of nitrate at different scan rate, 25 to 125 milli, micro per second. This is for this is the indication of sodium nitrate sensor. Sodium nitrate sensor at 0.4 millimole per. I forgot that one. Particular detection limit. So for sodium nitrate detections. Final conclusion, it is our compound CT8 AGNP will act as a multifunctional agent for towards antibacterial and anti-cancer activity, and as well as an electrochemical sensor analysis. Thank you. So thank you for the organizer for giving an opportunity to present our research work in this conference. Thank you, madam.
Thank
Any
you.
questions?
Dr. Subhendu Bhandari, now working as assistant professor in Department of Plastic and Polymer Engineering at the Maharashtra Institute of Technology, Aurangabad. Today, my topic is simultaneous toward doping with succinic and citric acid related toward synergistic improvement of electrical and thermal characteristics of polyaniline. By mistake, the last word polyaniline is missing from the title.
Yes.
First of all, let me discuss with you brief background and motive of the work. Polyaniline is a conducting polymer which can be synthesized in different ways in different routes, with different microstructures as well as in nanostructures, and its electroactive characteristics can be tuned by use of different types of acid dopants, as well as its as well as in combination. So far, different types of combinations was used and in our previous work, for the first time, we have come up with some interesting result with exhibiting synergistic improvement in terms of electrical conductivity, dielectric characteristics, as well as in electrochemical characteristics. Today, we shall not discuss about electrochemical characteristics. This work is that in previous all types of synergistic improvement, it was observed only with combination of the acids where one acid was weak acid and another acid was strong acid. So far, we have tried with some combinations of other types of acids also, and this is the first of work where we have come up with synergistic improvement even with the combination of weak and weak acids, both relatively weak acids. So in previous work, its synergistic improvement could be obtained in such extent where it could be it could be almost similar to the application of CNT to 4 or 8% level. In this work, we have first prepared aniline sulfate from aniline and sulfuric acid. Then we have followed polymerization method where mostly we shall not use any different type of organic solvent
Sir, you are not audible.
So I'm not
You-
audible.
No, now it's okay.
Now it is okay. Okay. So this slide, I was audible from experimental. Okay.
Yes.
In experimental part, we have started with preparation of aniline sulfate from aniline and sulfuric acid, and followed by aniline acid sulfate. All were used in solid state polymerization method without using any organic solvent to reduce the hazards of potential hazards of solvents in the synthesis process. We have used one acid, citric acid, which is tribasic acid, another acid, succinic acid, which is a dibasic acid, in different molar ratios, 0:100. That means fully used citric acid, that is 0.4, and succinic acid, all succinic acid, that is AC40, and intermediate, these three, 1, 3, 2, 2, and 3, 1 are the ratios of succinic to citric acid molarity. Total molarity was remained constant. In morphological analysis through ACM, we have observed that in succinic acid doped polyaniline, this is the individually doped aniline with succinic acid and citric acid. In case of succinic acid, mostly nanoplatelets were formed. In case of citric acid doped polyaniline, mostly small spherical nanoparticles were formed. In case of succinic acid to citric acid, ratio 1:3. In that case, some platelets along with some spherical particles were formed, but in the other two cases of dual doped polyaniline, mostly particle in the shape of mostly in the shape of spherical particles were formed. FTIR spectra, all the FTIR spectra of all the samples, it proved the presence of benzenoid and quinonoid, these two peaks, which is expected in case of polyaniline. Benzenoid for reduced structure, quinonoid for oxidized structure, and in partially oxidized and partially reduced form, both are expected, which gives the structure of emeraldine, structure of polyaniline, which is the conductive form. So from here we can observe that both the presence of both benzenoid and quinonoid structure proves the conductive structure. However, with the change of succinic acid to citric acid dopant ratio, we have observed to quinonoid ratio increased. With the increase of succinic acid concentration, benzenoid structure was increased compared to the quinonoid structure. And from XRD analysis, we have observed that with the increase of succinic acid concentration, crystallinity decreased. In dielectric characteristic analysis, dielectric was observed to be the highest in case of succinic to citric acid concentration, use of succinic to citric acid dopant concentration 3:1, that exhibited the highest dielectric loss as well as in the highest dielectric permittivity. However, as the ratio of the loss to permittivity, the dissipation factor was observed the highest in case of both these two in dual doped. The green one is 3:1 and the red one is 1:3. And the other sample equal molar use of two dopants resulted in the intermediate range of dissipation factor for the case of 10 delta. Both Cole plot and AC conductivity were in the same trend. It followed the same trend, where we have observed that synergistic improvement was observed in the case of succinic to citric acid ratio 3:1, where it exhibited AC conductivity higher compared to all other samples. And in the case of Kolkol plot also, we have observed the for the case of succinic to citric acid 3:1. Also, all the samples were found frequency independent. There was almost no effect of frequency in AC conductivity, which is a good characteristic of conducting polymer. This is the schematic diagram of the possible mechanism what we have proposed for our earlier work, and we have found that it also works. It is also applicable here also, where the relative change of some polarity of these two dopant ions, different dopant ions, dissociated dopant ions, and that helps in more conduction of current through the along the polymer chain. UV visible spectroscopy was were analyzed, where optical band gap were calculated from the top plot. It was observed that for the case of all these three dual doped samples, the optical band gap was less than the individually doped samples, where the 3:1 again, that this sample, again this green sample, succinic to citric acid 3:1 ratio, that exhibits the lowest optical band gap 2.85, which is much less than doped samples. In thermogravimetric analysis, these characteristics we have observed first. In this case, we have not observed such type of synergistic improvement in case of thermal stability. In case of any other combination, any other dopant combinations earlier. In this case, we have observed that all these three samples, these blue, green, and red, these three samples are dual doped samples. Samples exhibited improved thermal stability compared to the individually doped samples. And surprisingly, equal proportion of the use of succinic and citric acid that exhibited the highest improvement in the synergistic improvement in terms of thermal degradation. We have also performed the thermal degradation kinetics, but for time constraint, I am not showing here. There also, it was found that the thermal degradation also proved that.
Subhendu, sorry to interrupt
yes.
you. You have only 3 minutes.
Yes, this is the last one, and the conclusion part is remaining. So, from this work, we have we can conclude that the equal molar concentration use of combination use of succinic and citric acid, it improved the thermal stability, as well as the 1:3 and 3:1 ratio of succinic and citric acid dopant combination improved the dielectric characteristics, dielectric constant, and as well as conductivity, electrical conductivity, as well as it exhibited the lowering of optical band gap in case of dual doped samples. That's all about my work, and I must acknowledge my students group, Rushikesh, Prathamesh, Sandeep, and from IIT Roorkee, Uday Shankar and Dr. Anusha Bandyopadhyay, some facilities as well as Vijayalakshmi Vision and Professor Deepak Kastege from IIT Kharagpur for extending the electrical characterization instruments. And this is my contact ID for any possible collaboration.
There are some reports of that. One of the report is the World Water Air Organization report. In was out almost 60% of the world population is facing the problem of water scarcity. Now, as far as India is concerned, 54% of Indians they are also facing the problem of high to extremely high water stress. Now, just to overcome these problems, our government has taken various initiatives like Namami Gange and some other initiatives, but they are working on that. And recently, in 2023, our Prime Minister that also gave a special emphasis on the circular economy in the field of water conservation. And according to him, that Honorable Prime Minister informed that the government has laid a lot of emphasis on the circular economy in this budget. And it's well said that when treated water is reused, fresh water is conserved. It is benefit for the entire ecosystem. So there is a need to recycle the water, to reuse the water, just to come over the problem of this water scarcity. Now there are some reports that you can say that in Chennai that is almost
Slide is not moving, Professor.
again
Yes, slide is not moving.
earlier it's moving or not, ma'am?
Not slide is not moving. You just no, yes, now it has changed.
Okay. Now that's one of the report that is recently in 2024 that the Chennai is the city which is where there is no underground water fit for use. And now it's slides are visible or not?
Yes, yes, visible
Okay.
and moving.
Okay.
Please continue.
Okay. But the problem is why we are not reusing the water. One of the main reason is because the water contains various contaminants, and basically there are two types of contaminants. One which we call them and conventional, another that is the emerging contaminants. And conventional that dye and heavy metals, and emerging contaminants like other waste, pharmaceutical waste, microbes, pesticide, insecticides, herbicide, or some PCB product, that's a personal care product. Now, because of that presence in toxicity in water, that create an alarming scenario in our country. That we are take one or two examples. That one that a very well-known train that is run from Batinda and reaches to Bikaner, and that is known as commonly known as Punjab Cancer train. That is 60% of patients they are suffering from the problem of cancer. Now, there is a village in Punjab that is Shersawala, that every house, every home that have patient, that cancer patient. And why the Punjab is suffering from that very high cancer rate? One of the reason is that they are consuming large amount of pesticide. The another one is they don't have any, they have the little choice to drink highly polluted water, and that may be one of the reasons for that this problem, that is cancer. And recently there's a report in Times of India that is on 28 May of 2023 that 11 persons were killed because of that, amalgamation of that cocktail of sewage and illegally dumped industrial dye effluent, and that is responsible for the death of 11 people. And one more report is there of that is of CPCB, that is Center Pollution Control Board, and in that report there are 319.
Sorry to interrupt you, Dr. Surendra.
Ji ma'am.
Sir, your whole picture is blurred.
All picture that is this slides are also blurred, madam.
No. Slides are good enough.
Okay. Maybe some camera issue, madam. That maybe the problem with that laptop. I think slides are okay.
Yeah, slides are good. Okay, carry on.
Okay. Now there's a report of the CPCB. That's 2021, and as per the report of CPCB, that is there are 1,277 GPI industries, and out of that 319, if you talk about the Haryana, they are the textile industry, and GPI industries are basically which dumped almost 1 lakh liter contaminated water in that river. In basically Haryana, if you talk about the Haryana, it's in Yamuna. Now, out of these two contaminants, the major one is the dye and heavy metal. But if you talk about that, the major contamination part, that is the dye one, because dyes that's annual production is more than 7 to 10 to 5 tons and became a patent problem. And out of that, the total dye consumption in textile industry, 10 to 15% of them are being discharged as a waste into the environment. And one more thing is that to process 1 kilogram of that dye, we require 100 liter of water. And accordingly, that colored water that we have to discharge in the river. And if you talk about total annual production of dye, out of that, 54% of the total dye that we have to use in the textile. Now, that is a great impact on, even though that is not a good or a bad impact on the health or the aquatic system on the presence of these dye contaminants in the water, that it may create aesthetic problem, that poor sunlight penetration, that is very, that's not good for the aquatic life. And affect various organs of the human. There are so many, you can say, that problem that may create that is created by the presence of dye contaminants in the water. Now, as per our Prime Minister said that there is a need to recycle or to reuse the water, and but problem is why we are not reusing the water, because it contains various contaminants. Now there is need to remove these contaminants from the water, especially the colored contaminants from the water. Various techniques were developed for this purpose, like reverse osmosis, that all we know that we are using RO at our home, nanofiltration, adsorption, ion exchange, and the electrodialysis. But if you compare these three techniques in terms of total cost, power, and the water recovery, if the best technique is the adsorption technique, although it's not in that's used for the large scale, but if you compare these three criteria, that's the cost effectiveness and the power that is required for this purpose and the water recovery, if you compare all these three parameters with all these technique like reverse osmosis, nanofiltration, and ion exchange, electrodialysis, and adsorption technique is found to the best one. And if you compare all these all these five techniques, the adsorption, that all we know, it's the simplest process, that the adsorbate molecule, they get absorbed on the surface of the adsorbent. And why the out of that we go for this bioabsorbent for this purpose. Why bioabsorbent? They are cost effective, and generally in that case we are using the agriculture waste, and that's also good for the environment. Now you can use or we can use for this purpose water purification as a biomass as such, or it can be modified. It can be modified by physical modification, like all these cutting, grinding, milling, and by chemical modification, by treatment with acid and base, just to make it functionalize, some biological modification, and by forming a composite, grafting, and there are some, you can say that some modifications which are generally used for this purpose. Why biomass or its composite adsorbent? That's very clear, because they are of cost effectiveness, because of eco-friendliness, and they can easily remove the various contaminants. And very
Sorry to
simple to.
interrupt you, Professor Surendra, sir, and you have only 4 minutes to complete the
Okay, ma'am.
presentation.
And first we write down some research review article on this, and they published in a very good journal with a very high impact factor. And these are the other biomass-based composite that generally used for this purpose: hydrogel, metal oxide, polymer composite, CNT, and matter organic framework. But when we are going through this whole process, there's a research gap, that you can say. That one thing, that is the most of studies are on the removal of a single contaminants, which is not applicable to real world effluent. Second one, lack of studies on the simultaneous removal of cationic and anionic dye. There are so many reports in the literature which remove either cationic or mixture of cationic dye, or anionic or mixture of anionic dye. And these dyes, they are removed generally at different pH. But there are no report available in the literature regarding that removal of both the dyes or the mixture at neutral pH. The another one is the stability and safety of the adsorbent. They are not well addressed. And now, looking at all these, there's a need of a waste biomass. Why I'm using the word waste? Because the just to make the process cost effective and eco-friendly, which can be modified in a greener way and can be used for the removal of not only single contaminants, for a multiple contaminants, and simultaneously from the wastewater. And one other thing is, because we want to do all the study at neutral pH. Reason being, if you want to change the pH, that may disturb the aquatic life. And in that case, we go for certain some composed biomass which is not explored much, and for that we go for saccharum munja. Saccharum munja, that all we know that we use this particular biomass for making culm, or by making used to make some decorative material at home. But nowadays, because of that globalization, the use of this particular plant is decreased drastically, and it is considered now as a weed, and the farmer, they don't have any option, they have to burn it, and when they burn it, that may create a pollution. That is the biomass we have chosen for that purpose. The another one is just by going all that report, the CNT, carbon nanotubes, are the emerging potential solution due to the this, because of their nature, that is their cylindrical structure, high surface area, and they are thermodynamically stable. And the another one is we can easily carry out their modification. Now, that is a scheme that we used to prepare this composite and that material which we use for the wastewater purification. First, we take this saccharum munja, and then
Sir, sorry to interrupt you, Professor Surender Kumar. Did you done any work, experimental work?
Yeah. I'm just coming experimental, ma'am.
Sir, you have only 2 minutes.
Okay, I will just complete in 2 minutes. Fine.
Okay.
That's that you look at a slide. That is a saccharum munja comes, that we have peel off it, then we would go through the process, dyeing, grinding, and sieving. On one side, and on the side, we take this carbon nanotube, we carry out their functionalization by dipping into that sulfuric acid, nitric acid. Now we get both. After drying and sieving that the saccharum munja, and one is the functionalized carbon nanotube, then we made different composites with the help of a sonicator by using 0.25 gram of this CNT, then 0.05 and 0.1. Actually, we made 3 composites with 0.5%, 1%, and 2%. And when after optimization, we carry out the study. That's another plant that we use for the same purpose. That is the penicillium glucum. And again, we peel off, and after shaving and after function of this multi-walled carbon nanotubes, again we make these 3 composites. And after optimization of different parameters like pH, that concentration that we have to use in the starting, the doses, and then the contact time and particle size. After doing the optimization with the percentage removal, what we conclude that there's a 96% of removal for initial concentration 30 ppm, and we use that 2% of 1% of CNT with saccharum munja. And we also carry out all this, the thermodynamic studies, that there is an isotherm that confirm it's a multi-layer adsorption, and that kind of spontaneous, and that's a physioption as well. These are the different results of the adsorption model, and that best result whether you obtained with 1% of CNT with saccharum munja. That's about the kinetic study, and that results what? That's a pseudo second order kinetics, and that equilibrium was reached within 10 minutes, and it's spontaneous, physical, and endothermic in nature. These are some results of adsorption 1. And as far as the mechanism concerned, basically that involves the hydrogen bonding, pi-pi interaction, and that electrostatic interaction. These are the different type of interaction that involves between the adsorbate and the adsorbent. And these are the evidences of that mechanism.
Come to the conclusion, sir.
Okay, madam. One more thing, that we also have carried out some desorption studies, because we want to make the process that completely eco-friendly. And even after that, you just look at that. Even after 10 cycles, the material that have 95% or more than that.
Conclusion, sir. Conclusion, please.
And okay.
Conclusion, sir.
Okay, madam. That after doing all that studies, that we have prepared a material which can be used, that we can say, for the removal of more than multi components and all different type of dyes, that is cationic and anionic, at the mixture at neutral pH. And we have also that published in patent, Indian patent, and They have granted the Indian patent for this work.
Okay.
And that is one more thing.
Yes.
That's our team that involved in that, that in our research, myself, Dr. Sonika, and one of my student, that is Aruna, is in scientist in DRDO. Then there's Sarita, that Nishita, and my other scholars, they're involved in that.
Okay.
That's practically that what we use do in the laboratory, that basically we have a setup in the laboratory regarding that. We also tried comparing with that the purifier available in the market.
Sir, conclude, please. Sir, conclude it. Now it is last, I think.
That's all from my side, madam.
Okay. Thank you. Any query from the audience side?
Ma'am, I have a question to sir.
Yes.
Surender
Ji,
sir.
sir. Ji, sir.
Sir, please, just one question. And
Yeah, surely. I'll sum up it.